Hello, my name is Sherree Friend. I'm the product manager to Amnis, part of EMD Millipore. And today I'm going to talk to you about imaging flow cytometry, specifically about applications using the Amnis brand ImageStream X and flow imaging flow cytometers. First, I'll tell you about how it works, what the benefits of using the imaging flow cytometers are, and then go through a few applications in detail. I'll talk about cell signaling, nuclear translocation, internalization, looking at a model of virus uptake, colocalization, looking at intracellular trafficking, shape change, a monocyte chemokine response, apoptosis, looking at DNA fragmentation as compared with a couple of other methods, and cell interactions, finally looking at immunological synapses. The systems include the instrument, the ImageStream X Mark II, and the flow cyte, which captures several fluorescence images at high speed, also captures bright field and dark field side scatter imagery. The IDEA statistical image analysis software, which quantifies the image analysis, gives you a number to put on the morphology, as well as gives you population statistics. These address complex biological questions because they have spatial information of biomolecules that calculate cellular morphology as well as intensity. Here's a picture of the instruments, the flow cyte on the left and the ImageStream X Mark II on the right. They both operate under the same principles, the same engineering principles. The main differences are the footprint, the cost, and the resolution. Here's the ImageStream layout, and I'm going to go over the principles of operation using this diagram of the optic system. The cells are introduced into flow, much the same like a traditional flow cytometer. The difference is that the detector is a CCD camera. The cells in the flow cell are illuminated by bright field illumination and laser illumination. The fluorescence and the bright field imagery and side scatter are then captured through an objective lens and collimated to the spectral decomposition element. The spectral decomposition element is a series of long-pass filters set at precise angles such that the spectra is spread out across so that when it's sent to the CCD camera, the light is captured in discrete locations on the camera, different wavelengths in different channels, spread across, in this example here, 6 channels. The camera is a custom camera and it's operated in time-delayed integration mode, which I'll talk about in a minute. We also have exact spatial registry of those images from one channel to the next, so that every pixel is exactly the same location from one place to one channel to the next, so that they can be perfectly overlaid, so that you can do things like look at colocalization of probes and location, specific location within the cell of those probes. This is what the imagery looks like as it's running on the instrument. This is an example of 6 images captured for every cell. So one row is one cell. The side scatter is in the column 1 in this example, bright field in column 2, and in column 3 you'll see a FITSI NFkB image. You see that it's inside the cell, and then 2 surface stains in orange and red. And then finally, a pink DRAC5 image of the nucleus. The ImageStream layout, when you have a fully equipped system, has 2 cameras, giving you 12 images per cell. The layout here is also, the lasers on the bottom of this slide are pointed to 2 different cameras. So some of the lasers are pointed to camera 1, and some of the lasers are pointed to camera 2. This gives you the ability to produce 12 images with multiple fluors. It gives you a large range of fluorophores that you can use. The objective lens on the ImageStream can be upgraded to include not only the 40x magnification, but also the 20x and 60x magnification. The 20x objective is useful to give you a wider field of view for large cells. The 60x is valuable to give you a higher resolution. And the 40x is the standard magnification that comes with the instrument. All of these lasers are variable power. They're adjustable, which allows you to adjust the sensitivity of the experiment. The flow site layout is a little bit different. The flow site uses one camera, and instead of spreading those 12 channels out over 2 cameras, we use one camera. 6 channels are on one quadrant of the camera and 6 channels on another quadrant. The flow site also uses a 20x magnification and has fewer options for the lasers. This allows us to bring the cost down on the instrument. So let me talk about time-delayed integration for a minute. The way this works is as the cell or object is in front of the detector, the CCD camera, it's illuminated over the entire length. And as the cell is entering the flow cell and is in front of that first pixel row on the camera, the light starts to become integrated and is captured in that top row. The cell's moving. As the cell is moving, the light that's captured in that pixel row is integrated into the next pixel row in exact synchrony with the flow rate of the cell, such that by the time the cell reaches the bottom of the camera, you can see it's getting brighter and brighter. We've integrated that light over the entire length of the camera, and the image is read out in the bottom row and reconstructed. So I'll show you that one more time. The images, therefore, do not streak or blur, and we get very high photonic sensitivity. This is the proprietary Amnis technology. The instrument's user interface is very intuitive, easy to use. There's several portions to the interface. The top, as you see here, is the image gallery. Every cell coming through is imaged in flow. And you can see here there's 12 channels across, and there are a couple of bright field channels, and the rest are fluorescence channels and a side scatter channel. So you'll be able to see what's coming through real time. In addition, there's a workspace where you draw graphs and gate on populations. You can collect any population you would like. You can do compensation on the fly while you're collecting your data, or you can collect it uncompensated and compensate in the analysis software later. The instrument controls are on the right. They're very simple, intuitive. This is where you load samples. You can also return what isn't run through, so that's a nice feature to get your samples back. You set your file acquisition parameters here. You turn the lasers on and off, adjust their power to adjust the sensitivity for the experiment. So everything is done on this front panel. The way the system is designed is so that there's automated calibration and tests that you run every morning when you start up. It takes about 15 minutes to get the system going, and it's all automated and calibrated. And if there's any failures, you'll see a red flag. This is done using beads. In the case of the ImageStream, it's a reagent called speed beads, which is run during all of your samples as well to control for that flow speed that I talked about when we talked about time-delayed integration. Also, with the flow site, you use a calibration bead. It's not run during the run, but that's something you do at the beginning of the run. All of the results of the calibrations and tests are logged in a history database where that can be pulled up at any time. It's a very nice record of your quality control for the instrument. I'd like to talk a little bit about the benefits of using imaging flow cytometry. Imaging flow cytometry gives you the best of both worlds. It combines the speed, sensitivity, and phenotyping abilities of the flow cytometer with the detailed imagery and functional analysis that you get with a microscope. Here you see some traditional flow cytometry data, intensity plots. Below you see people hard at work looking through the microscope. With the imaging flow cytometer, you can collect many, many events very rapidly and get both types of data, flow cytometry data and imaging microscopy data. But when you do that, you can actually do things in addition to either one alone. The imaging flow cytometer does not sacrifice sensitivity. The fluorescence sensitivity of the imaging flow cytometers exceeds that of most conventional cytometers. It's as good as the best. Higher absolute sensitivity allows detection of dim cell markers. The uniform sensitivity eases panel design. Even red dyes are very bright. As you can see in the plot on the left, this is an example of 8 peak beads. This happens to be run on the flow site. We have very low coefficient of variations, or CVs, very tight peaks. You can see all 8 peaks very well, even out in the red channel. What this does is it allows you to resolve populations in mixed cell population, in mixed cultures, or in blood, for example. Here's an example from a traditional flow cytometer where you get very good separation of using CD45 only in side scatter of lymphocytes, monocytes, and granulocytes. What that looks like on the imaging flow cytometer is this. You can get a 5-part differential, even picking out those dim basophils and the bright eosinophils, just with CD45 and side scatter. If you click on the dot in the eosinophil population, every dot on this graph is connected to its image. So if you click on a dot, you'll be able to see that image for that particular data point. Click on several dots. It allows you to verify the populations. It allows you to gate with confidence. Once you have your gate set, you can then check and make sure that you gated properly and you see your imagery. You can also show populations of these cells and verify all of this imagery. Here you can see the eosinophils have that nice bilobe nucleus. The neutrophils are multilobe, and that bright CD45 lymphocyte population with those round, small nuclei. In addition, you can calculate morphological parameters. This is an example of calculating lobe count. Again, as I mentioned, in the neutrophils, you can see that there's multiple lobes, and you can count the lobes. This is quantifying those lobes. And you can look at the lymphocytes and see that they're mainly one lobe. Some of them have a different shape. Because you can get that morphological information, you get much more out of your data. Here is how people have been using these imaging flow cytometers and publishing data over the last few years, over the last 10 years. From the first publication in 2004 to this year, the number of publications has grown year over year. And in some cases, high-impact journals have chosen to put ImageStream data on the covers. There's a wide range of publications. There's unlimited potential here. On the left, the publications by research applications is shown. You can see that the predominant publication is in cell signaling area. There's a lot of data in looking at nuclear localization, which I'll talk about in a minute. Internalization, morphology analysis, as well as a large chunk of emerging applications. So people are continuing to discover new applications using the imaging flow cytometers. On the right is a graph showing the disciplines. It's been mainly used in the immunology field. That's probably because immunologists have been using flow cytometers for many, many years. But there's a large chunk of biochemistry that's done on the instrument. Looking at your proteins and your RNA and your DNA and where those proteins are is very easy when you have the image. And you can do that on thousands of cells in a population basis. Oncology, drug discovery, hematology, virology, there's many, many applications here, and we're still discovering places where the instrument is being used. So let's talk about some applications. This is a list of imaging flow cytometry applications that have been done. It's by no means complete. But you can see that there's beautiful images here for every one of those applications in cell signaling, which I will talk about in a minute, shape change or chemotaxis, internalization and colocalization, cell interactions, immunological synapse interactions, and finally, cell death and autophagy are the ones that I'm going to point out in detail. I won't have time to talk about all of them, unfortunately. So let's talk about cell signaling. This is taken from that publication list that I showed you a minute ago, this is taken from the titles. And what I want to point out here is that when you're talking about cell signaling, we're talking about not just looking at nuclear localization, but we're looking at RNA and proteins, different transcription factors, looking at, for example, the IRF5 translocation in systemic lupus erythematosus, one of the autoimmune diseases. We're also looking at human ovarian tumors and how signaling occurs in those lymphocytes that are associated with those tumors. People have used the instrument to look at FOXO1, at NFkB, at NFAT, JNK, also looking at EGF clustering. So there are many, many different types of biochemical cell signaling applications. As an example today, what I want to talk about is looking at NFkB. So NFkB is one of the proteins that is activated from many different cellular activation processes. The signaling from the outside of the cell going through into the cytoplasm, this cascade of events that happens that culminates in the interaction of the NFkB and IkB, that phosphorylation event of the NFkB, then releases it to be moved into the nucleus, then binding to the DNA and activating the cell, and then all the downstream events that happen. The NFkB is one of the most published of our applications. How is that done in conventional ways? Molecular analysis using Western blots or gel shift assays on bulk populations. Whether you've isolated a pot of cells or it's a cell line, it's a group of cells, it's a population of cells that you're analyzing. And if you have a band, then in the nucleus or cytoplasm, this is you're measuring a pot. Or immunofluorescence assays using microscopy or flow cytometry. With flow cytometry, that's generally done with a phosphoprotein, so looking at the antibody specific for that phosphorylation event. On the microscope, you'd be looking at images, and in this example here, this is NFkB in green and the nucleus in red, and looking at where that protein is. Is it out in the cytoplasm or in the nucleus? And that's a very objective, low throughput assay on the microscope, even though it's single cells, and you get a lot of information about that cell. So let's talk about the Western blot first. There's several outcomes from that Western blot that could happen. When you have complete translocation of all your cells, you're going to get a very dark band. When you have no translocation in any of your cells, then that band there's going to be no band. If you have partial translocation, there's a couple of different outcomes that could happen. You could have 50% of your cells translocating all of their NFkB into the nucleus and get a dim band, or you could have 100% of your cells translocating half of their NFkB into the nucleus and get that same dim band. It's hard to tell what the difference is on the individual cells with that outcome. And now let's look at microscopy. So this is an example of stimulated monocytes using LPS, and it's incomplete, so it's an intermediate dose. Again, NFkB is in green here, and then the nucleus was stained with DAPI, so it's this bright purple pseudocolor. And I think everybody here would agree that the 3 cells circled in green all have their NFkB in the cytoplasm, have not translocated it into the nucleus. And I think we'd all agree on that. And we'd probably all agree that those 2 cells circled in red have all of their NFkB in the nucleus, and those 2 cells have completely translocated. So we would call those translocated. But what about the rest of the cells? I think we might get into some arguments over, is this one 75%, is this one 25%? It's very hard to quantitate using microscopy, and it's objective. I'm sorry, it's subjective. So I'm going to show you an example of NFkB translocation in whole blood subsets. Here we've incubated whole blood with LPS to stimulate the cells. Then immunophenotyped the leukocytes and stained for NFkB and the nucleus. Collected 20,000 events on the ImageStream or Flow Cyte, and then quantify nuclear localization in the subsets. This is the immunophenotyping step where we've used antibodies and scattering to identify the eosinophils, the monocytes, the T cells, and the neutrophils. Once you have your immunophenotype and then you stain for NFkB, you can measure the NFkB translocation by the staining of the NFkB image here in green and the DRAC5 image here in pink. The cell on the left, as I described a minute ago in the microscope data, is untranslocated. You can see that the NFkB is predominantly in the cytoplasm and there's very little in the nucleus. And the cell on the right is translocated, where almost all of the NFkB is in the nucleus. And at the top, you can see that they overlay very nicely. The way this is calculated, we use a feature called Similarity. And what Similarity is the Pearson's correlation coefficient for the pixel by pixel location of the NFkB staining and the DAPI staining. So on the cell on the left, if you take the NFkB intensity for each pixel versus the DAPI intensity for each pixel, you can see that as the DAPI intensity gets high, the NFkB intensity is getting low. Therefore, we have a negative slope. We have a negative correlation. There's a very low similarity score. The value 2 is the fit to that line. The cell on the right has a positive correlation. As the DAPI intensity is getting brighter in a pixel, so is the NFkB. So that quantifies the co-location of the NFkB and the DAPI. And this cell had a very high positive score, highly translocated. If we look at a population of cells in this intermediate stimulated sample of LPS, you can see that there's a variety of similarity scores. You have a wide spread. Each of these cell images has been taken from the bin with that similarity score. So in the upper left on the slide, you can see that negative 2.2, very low similarity, very not translocated. If you follow this around under the graph and up to the top right on the slide, you can see that cell has a very high correlation, positive 4. All of the NFkB is in the nucleus. But you can also quantify all those intermediate steps. It's very sensitive, very quantitative way to measure NFkB translocation. When we go back and look at those subsets that we analyzed by the leukocytes phenotyping, you can see that in this unstimulated sample, all of the populations have low similarity scores. The mean of these populations is low. In the stimulated sample, you can see that the monocyte population has shifted to the right. We have a very high similarity score. So then we can make the conclusion that it's the monocytes in this population that have translocated their NFkB into the nucleus, not the T cells, the eosinophils, or the neutrophils. Let me talk about internalization applications now. Here's a list from the titles of some of the publications where people have published internalization applications. People have looked at macrophages taking up Candida, phagocytosis, looking at both Helicobacter and actually taking up apoptotic epithelial cells. Looking at rituximab entering into B cells, looking at microparticles in dendritic cells, nanoparticles containing PD-L1 in Kupffer cells. It's all over the place. There's lots and lots of data on internalization, one of the major applications for imaging flow cytometry. I'm going to show you an example that was done by Dr. Patricia Fitzgerald-Bocarsley at University of Medicine and Dental School in New Jersey. She graciously let us use this data. Dr. Bocarsley is interested in virus uptake, and as a model is using CpGB labeled and incubating that with peripheral blood mononuclear cells in order to look at the trafficking of this uptake by plasmacytoid dendritic cells in whole blood. So here you see the CpGB molecule that's labeled in red, and looking at the plasmacytoid dendritic cells labeled with BDCA1 in orange on the cell surface. Also staining for the internal organelles, the endosomes and the lysosomes, with either CD71 or CD107A in green. We collected 20,000 event image files on the ImageStream for this because these plasmacytoid dendritic cells are a rare event in whole blood. Then quantified the internalization and colocalization to the endosomes and lysosomes. So here is, on this graph, the CpG that has been incubated with the peripheral blood for 30 minutes at 37 degrees. And on the x-axis, we measure similarity bright detail feature, which is used to measure the colocalization. And what that means is we're looking at the bright details of this staining because the CpG molecule has a very bright punctate staining, and so do the endosomes and lysosomes. So when we look at the colocalization of those, we're looking at just that detail, which makes this a very robust assay. On the y-axis is internalization score. So anything above 0 that's positive, we are calling that internalized. Half the intensity is inside the ring of the BDCA and half is outside. That's our cutoff in this example. So let's look at the lower left quadrant here. These are the images that are in that quadrant where you can see that the CpG molecule is actually on the surface of these cells. And in this case, the CD107 is on the inside, which is as it should be. In the upper left quadrant, you can see that now the CpG molecule has entered the cell. It's inside that ring of the BDCA positive stained cells, the plasmacytoid dendritic cells, but it has not yet colocalized to the organelles, the lysosomes in this case. And then in the upper right, you can see that now the CpGs actually entered into the lysosomes, and there's an overlay, and there's a very high bright detail similarity score for these cells. They did this also as staining with CD71, looking at the endosomes, and here's the time course. So as a control, incubated on ice so that the CpG molecule was minimally binding to the plasmacytoid dendritic cells, you can see there are fewer cells in that plot. At 30 minutes, about 24% are colocalized into the endosomes, and at 120 minutes, about 20%. So it's going down over time. That's because they have now colocalized into the lysosomes, a later time point in the incubation, where we're going from 27% at 30 minutes to 67% at 120 minutes. And so you can look at trafficking like this. Over time, the CpG molecule is going into the endosomes and then moving into the lysosomes. The next application I'm going to talk about is apoptosis. So on the imaging flow cytometer, you can measure apoptosis by looking at DNA fragmentation. That's the hallmark of apoptosis. Programmed cell death, or apoptosis, causes DNA to fragment and becomes smaller in the nucleus and punctate staining. To compare that with some other methods, we're going to look at also caspase activation and annexin 5 binding. So in this example, we incubated Ramos cells with 5 micromolar camptothecin, which induces apoptosis. It's a very large dose. This is an example of the bright field image, and you'll notice that the contrast is very high here on a cell that is apoptotic, which I'll come back to in a minute. The cells are labeled with Lux, which binds to active caspase, annexin 5, which binds to the phosphatidylserine, which is flipped from the inner leaflet of the membrane to the outer when apoptosis is happening. And we also labeled the nucleus, in this case with DRAC5. And we collected 20,000 events to analyze for these 3 ways of looking at apoptosis. This slide's a little bit busy, so let me walk you through it. On the x-axis is the Lux intensity, or the active caspase, and on the y-axis is the annexin 5 intensity. You can see that 67% of the cells are double positive for this. But there are a few cells, 4%, that are stained with the Lux for the active caspase, but they're negative for the annexin 5. Whereas there's about 10% that have bound the annexin 5, but are negative for the Lux, or the caspase activity. Together, at most, you could say probably 77% of these cells are apoptotic. However, if you look in the lower left quadrant, there are cells in that quadrant as well that have fragmented DNA. So we've missed some with both of those assays. The way we measure apoptosis on the ImageStream or the flow cyte is generally looking at the area of the nucleus. When the area of the nucleus is small, that means the DNA has fragmented and the area is very small. We also can measure the contrast in bright field. So the cells on the left here are from the white dots in the upper left of the left graph, which is an untreated sample. And you can see that the nucleus is very large. There's an even staining of that DRAC5. The bright field contrast looks very low. And on the right is the camptothecin treated sample. You can see that 90% of those cells have fragmented nuclei and have very high bright field contrast. So we've captured all of the apoptotic cells in this assay using just the DNA stain. Lots of times you can multiplex assays and get apoptosis for free if you just add DNA dye with your other assays. Here's a graph comparing the 3 just as a conclusion over time. So it's time, here's a time course confirming the differences, and you can see that over time you can look at the annexin 5 goes down, the Lux is coming up and staying the same, but the DNA fragmentation obviously is capturing everybody. Okay. The next application I want to talk about is looking at shape change and in terms of chemotaxis. I'm going to show you an example of looking at monocyte population, but I wanted to show you that there was a publication just last year looking at T-cell shape change. This is a publication from the FDA where they were looking at a heparin contamination event that happened to be this oversulfated chondroitin sulfate, and this helped them to screen those heparin samples. So what we did was we looked at human PBMCs in response to MCP1. MCP1 is a chemokine that attracts monocytes out of the bloodstream into the interstitial spaces in response to an inflammatory event. The monocytes have to extravasate through the endothelium and get into those interstitial spaces, and therefore they have to reorganize their actin and change shape. So we'll go from that nice round cell to this cell that doesn't have that nice round shape any longer. We stained the membrane with CD14. This assay can be done with the membrane stain or the bright field image, however. And we collected 5,000 events on the ImageStream or Flow Cyte. And here's the images showing that. Then we quantified the monocyte shape change using a feature called circularity. That feature measures the variance in the radius of the cell. So when you have a high radial variance, then your cell is no longer round. Here's the graph of the circularity for the untreated and the treated sample. The graph on the left, you can see that 9.5% are in that low circularity region called R2, whereas on the right, the MCP1 treated sample, there's about 61% of those cells that have low circularity. And what do those look like? Well, the cells in R2 on the left graph, you can see there are some monocytes that are not perfectly round, but the majority have this nice round circular morphology. And the treated sample, those 61% look like this. You can see they're not round anymore. They're moving around. They're rearranged their form in response to the MCP1. Now I want to talk about applications in cell interactions. And I'm going to talk about immunological synapse specifically. There's a few seminal publications that I have listed here on this slide, and there have been many more very recently published. We did this experiment at Amnis where we took T cells and APCs and mixed them together with SEB, Staphylococcus aureus enterotoxin B, in order to stimulate human T cells. SEB binds to a particular V beta molecule on T cells that are on a large number of T cells. And so this will give you a nice response. It's also why you get sick at a picnic if you eat the mayonnaise that's been sitting out all day. What we did here was we took those antigen-presenting cells and we incubate them with the enterotoxin, mix them with the T cells, and incubate them together. And then fix and stain, and I won't go through those details, but the stains that we used for this were CD3 to identify the T cells, and that's this orange cells here, CD19 to identify the antigen-presenting cell, actin to look at the reorganization of the, phalloidin to look at the reorganization of the actin filaments. And then we also did a couple of experiments where we looked at an integrin molecule, LFA1, which moves to the T cell receptor, help organize and bind the T cell receptor to its antigen-presenting cell. LCK, one of the signaling molecules that is recruited to that in the T cell to that site where the synapse is going on in order to translocate that signal through to the cascade all the way down to the nucleus, where NFkB is stimulated to move into the nucleus. So we also stain for NFkB, and we use DAPI to stain the nucleus. So I'm going to show you the strategy to analyze this data. This is the way we isolate one T cell and one APC in order to look at that, interrogate that immune synapse. First, we have to identify doublets. So there's a couple of morphological parameters that we use to do that. The area of the bright field image versus the aspect ratio of the bright field image. Because when you have an aspect ratio around 1, the cell is very round. Its width by height is about the same. So you're going to have an aspect ratio around 1. And you can see that the area of the T cells is smaller than the area of these large APCs. So you can even separate those if you wanted to. But what we're really interested here is the doublets. And you'll get doublets of T cells, you'll get doublets of APCs, you'll get doublets of an APC and a T cell, and what we want to do is get down to analyzing just one T cell and one APC interaction. So in order to do that, we first isolated the double positives, the CD19, CD3 positive cells, and then using that same morphology analysis, only this time on the stained image, so on the CD19 in this case, we looked at area of the CD19 image versus aspect ratio. And we can isolate those conjugates that only have one APC. And you'll notice here that a couple of them have a couple T cells associated. So then we go through the next step with the same morphological algorithms. However, this time we're looking at the CD3 image, so area versus aspect ratio of CD3, and now we're down to conjugates that are just one T cell and one APC. Now, when you're analyzing image data, there's one thing that you can do is look at a region of interest. In all of the applications that I've been talking about, we have been using masks sort of to look at the region of interest, whether it be the nucleus or whether it be the spots in the image or whether it be the cell surface. In this case, what we want to do is focus our attention on the contact between these two cells, the interface. So there are many masks in the software, in the IDEAS software. One of the masks is called interface, and we have another one called valley, and there are ways that you can focus your attention on that point of contact between the two cells and then measure what's in there. And as you can see in this example, the cell on the top has very little actin in that mask, behind that mask, which is shown as the blue overlay. The cell on the bottom has a very bright actin image, so it has a mature immune synapse. When you calculate those features in the mask, you can see that on the left here are the cells that are in contact, but there's not a mature synapse. You can see that the actin has not accumulated there. Whereas the cells on the right, we have a very mature formed synapse. You can see the actin is very concentrated right in that synapse. And we measure that in this graph. The pink dots are the treated sample, and the green dots, you probably can't see very many of them, are the untreated. Of course, the untreated has very fewer numbers of conjugates. The percentage of T cells that are associated with it have mature immune synapse in the sample with the SCP-treated sample was 5.5%, and then the control was 1%. It was a very statistically significant difference. Remember, we're looking at rare cells, rare events in this large population. We also looked at LFA1 migration to the immunological synapse. And in this case, we switched the colors around a little bit. The APC is now in yellow. The T cell is still orange. The actin is red, and we included the LFA1 in green. So these cells on the left are the cells that do not have a mature immunological synapse. The cells on the right did have a mature immunological synapse, and then we measured the amount of LFA1 at that synapse. And the graph below shows the differences between the control and the SCB-treated APCs. The SCB-treated APCs are in the red dots. Then we looked at Lck migration to the synapse. So Lck is a Src family tyrosine kinase that is involved in the T cell receptor complex. It helps to transduce that signal of activation into the cytoplasm through the cell. And in this case now, I'm going to tell you about the colors on the cells again. The T cells here are in yellow, the actin is in orange, and the Lck is in red. So you can see the cells on the left where there was no mature immunological synapse. There's very little Lck associated there with at the synapse. And the cells on the right, you can see the accumulation of Lck. And finally, looking at the final event, the activation of that T cell, this sample here is, again, the APCs are in orange and the T cells, the APCs are in yellow and the T cells are in orange. But then we added NFkB, which is in a green color, and the nucleus, which is in a sort of purple color. Here, these T cells are from the same tube. So we looked at the activation because we can focus down on just the T cell and measure the NFkB location in just the T cell. We looked at the free T cells that were not associated with APCs at all, and then those T cells that were associated with APCs and formed immunological synapses, and then measured the NFkB translocation. So the red line on this graph are the free T cells, and you can see they have a low similarity score. And the green line on this graph are those T cells that are associated with the APCs and have been activated, as evidenced by their NFkB translocation and that shift to the right. So in conclusion, today I told you about the ImageStream X Mark II and the Flow Cyte. They give you the best of both worlds. Fast, sensitive flow cytometry data with detailed location information of a microscope. This enables applications that neither the flow cytometer or the microscope can do alone. I presented data showing you the output from the IDEA statistical image analysis software, which quantifies image analysis and population statistics, puts a number on that morphology. This allows you to address complex biological questions like nuclear localization, internalization, cell interactions, apoptosis, and much more. So thank you for listening. If you'd like any more information or you'd like somebody to contact you, you can go to our website, emdmillipore.com/Amnis. And just remember, you never know what you're going to find in your cells. Thank you very much.